So do I advance it to get the first slide? Is there a control mechanism person? No. Well, this doesn't count as part of my time. Well, I could do it without slides, but I prefer to. Is there an IT guy that can, oh, well, let's go. So anyway, I'm going to give, Jim has done a wonderful job of introducing Ilya. So I'll talk about my personal interactions with Ilya. So I remember about 2005 going to Stu Kaufman, who's here, and saying, look, we'd really like to get a data scientist. Who would you recommend? And he said, there's this young guy at MD Anderson that's absolutely spectacular, Ilya Smolovich. And that was the only person he thought was of a quality that we should go after. I went down pretty quickly to MD Anderson and spent a day with Ilya. And it was utterly a marvelous day. I was impressed with his openness. I was impressed with his appreciation of a diversity of different kinds of things. And I was certainly really impressed with his science. As a consequence, he came to ISB and has been a foundation of ISB, just as we heard from Jim. When Ilya got sick, it started a series of conversations that we had, I would say, every 2 to 3 weeks, where we just, for roughly an hour, sit and talk about everything. And in fact, it was about science. It was about what's going on in areas he was interested in, music. But what was most interesting was the evolution of his attitude toward his disease. And in essence, he became very Buddhistic in being able to distance himself from the disease on the one hand. And I think Ilya actually was more effective at assuring people things were going to be okay than we were in assuring him. But the other thing that was remarkable is he didn't want to let it stop him from doing what he valued. He did his science, and one a really spectacular example is rather late in the disease, Ilya and his son bicycled from Seattle to Friday Harbor, which probably is a 100-mile bicycle. And Ilya was determined in all dimensions of his life to continue on. And I agree with Jim. I think in many ways, each of us at ISB felt we were very close to Ilya. He had this marvelous ability of drawing you in. Now, what I did is looked in my CV at the 4 papers I had with Ilya across the stage of his career. And the first, and each of them opened my eyes and impressed me in different ways. And the first was macrophages exhibiting criticality. We won't go into the details, but many of the people that are in this audience were on that paper, as a matter of fact. And it was the beginning of my thinking about biological information in a very different way. He was also willing to help with simple technical problems in analyzing genomes. One of the big problems in the early days was being able to be confident you could see little repeat segments that themselves did have biological importance. And he worked together with Gwen Gustavo and again, several other people that were here on this problem. The third paper was a paper that Theo actually played absolutely a major role in. We had an interrelationship over many years with a big hospital center in Virginia. And the basic question was, can we take preterm births and stratify them according to how early they are and find markers that can delineate those? And the basic answer was yes. And again, Theo, Ilya played really an interesting role in that. And then finally, this shows you what I said about not giving up. This is a 74, it's a 24 paper that just came out with Ilya. We were interested in doing a very large scale diabetes study. And I went to Ilya and said, would you help us assemble a digital twin for diabetes? And he did so. And this is the start of that effort and so forth. And in the final thing I'll say is Ilya really played an important role in this book on systems biology and medicine that Cold Spring Harbor is hopefully going to publish in the not too distant future, which talks about what systems biology is and how it can be applied to medicine. And I think what I'm most excited about in this context is I hope medical student courses will use this book in something that's utterly deficient in medical school teaching, namely understanding systems biology and its really fundamental applications to biology. So in that context, let me talk just a little bit about this Human Phenome Initiative. I set up in 22 a nonprofit called Phenome Health to drive the whole idea of this idea of data-driven health. And of course, that had to do with genome-phenome analysis. It had to do with analyzing the data, and it had to do with leading to actionable possibilities for individuals that could either improve their wellness or let them avoid disease. And that's kind of the short-term thing that we've been working on. And the long-term thing is to propose a second genome project, an extension of the first that looks at 1 million people over a period of 10 years with regard to these data. And I'll say a little bit more about the million-person project in just a moment. And of course, it was this company called Aerovel, a wellness company that existed from 2015 to 2019 that generated genome-phenome data from 5,000 people to varying lengths of time. And from these data have come more than 25 different papers. And they give us beautiful glimpses into the two fundamental dimensions of data-driven health. One is the ability to optimize wellness, and the second is the ability to detect very early disease and be in a position to reverse it before it becomes complicated. And I would argue that a really fundamental point about wellness and transitions to disease and transitions back is this really critical idea. And we actually had papers on 2 animal mouse models where we could induce disease. And as you'll see in a moment, we looked at 1 patient in the same regard. And the simple point is, at the earliest stages of the disease, long before clinically manifest, the changes are simple. And they become exponentially more complicated as time progresses and everything. So, and I'll tell you how data-driven health can see these transitions from 2 different points of view. So the 3 transitions here, and I'll talk about each of these in turn. We can really improve wellness. We can identify these transitions. And of course, we can use powerful tools to deal with extent of disease and everything. So data-driven health is about generating the actionable possibilities to improve wellness and metrics for assessing that. It's about the actionable possibilities to detect early chronic diseases to reverse. And it's about taking a big data and large population approach to understanding the complications of disease and so forth. And with regard to wellness, there are really two types, as we know now, traditional and the data-driven wellness. And the traditional give you general advice about how you can improve, deal with each of those four features. Whereas the data-driven wellness take the genome, and from that, there are at least 7 different categories of actionable possibilities, and they add up to more than 20,000 different possibilities now. So it's really striking. We can run through those if you wish. And from the Phenome, the same is true. We can take data from our clinical chemistries, and it says a great deal about many of the common diseases and gives you hints like LDL cholesterol about, if necessary, taking statins, but there are other options we'll talk about. So there are a whole series of things from the blood and the other phenomic measurements we can do. And most rich with regard to actionable possibilities is the integration of genome and phenome data. And an example of myself, I had in the initial study rock bottom levels of vitamin D, 100 international units a day, 1,000 international units a day, didn't touch it. And it turned out I had 2 genes that block the uptake of vitamin D, and you could circumvent that difficulty by taking 15,000 international units till I got to normal, and then backing off to 5,000 international units thereafter. But the important part is that with data-driven health, we can optimize each of those 4 features for individuals. And the really important part is data-driven wellness explodes well beyond the classic wellness. I would say the typical well person today out there has exhausted 20 or 30% of their potential. And the real question is, since you have to make the decisions, what choice do you want to make for this kind of thing? So metrics of wellness, biological age, came from studying people that range from 21 to 93. And I won't go into how we got the algorithm, but what proved its validation is the algorithm, when applied to the Airville population, showed that women lost 1.5 years of biological age per year in the program, and men lost 0.8 years in the program. So we use this as a fundamental metric at each blood analysis for wellness. Noah Rappaport really has pioneered this idea of a biological BMI as contrasted with BMI, the classical one using height and weight to make the measurements. It's flexible. It captures 30% of the population that the traditional one misses. It allows you to stratify things like obesity and so forth in very different ways. And it's instantly responsive to changes in diet and overall metabolic status and so forth. Noah, again, with colleagues, Alice Lane, and I think Sean Gibbons is working on a metric for frailty, and this that is independent from your age. And this measure of frailty is really an important metric for assessing how well you're aging as you go on, and when there are disastrous downturns that we can respond to. So these 3 metrics are going to be key. And a 4th one, again, which I won't go into, is we now have blood organ-specific proteins for about 25 different organs. And in one case, the liver, we followed in humans an infectious process, hepatitis, in the liver from before and after, and showed beautifully the 88 organ-specific proteins for the liver said, you have a change in state, and it pointed toward the kind of networks that are involved in that change of state and so forth. So the early detection of chronic disease Eve was the first one of individuals who in the program transitioned from wellness to disease. And what we simply did was to take the 6-month space blood draws prior to her clinical diagnosis and looked at metabolites and proteins in the blood. And we saw a series of 5 proteins that were modified at the earliest state we could see. And 3 of those 5 proteins mapped into disease-perturbed networks specific for stage 4 pancreatic cancer. And what we were able to do is, in looking at the overall 167 transitions to chronic disease in the population, 30 of them transitioned to chronic diseases, and we looked at the 10 that transitioned to various kinds of cancers. And for each of them, we found exactly the same kind of thing. So the really important point about this then is, one, that the proteins point toward networks that show you where you ought to go to start reversal of the early stage of disease. And they tell you about some of the things that are happening in the beginning of the disease. And number 2, with the million-person project, we calculate there'll be 200,000 transitions, and that'll give us enormous data for all chronic diseases and the subtypes of chronic diseases and the correlations we need to begin large-scale thinking about therapies that can reverse at this earliest stage. A key point is that in our 4.5 trillion healthcare budget, chronic diseases cost 86% of that. So if you can make significant impact on chronic diseases, you have monies freed up to go and point toward wellness and prevention. And in fact, Deloitte just published a couple of years ago a prognosis of this kind of thing and how big a change in the next 5 years there would be in this regard. Now, the final point I'll make about Eve, which is really striking, is we looked at 9 disease-perturbed networks in her stage 4 pancreatic cancer. And what we were able to beautifully demonstrate at the various blood draws is, in the first blood draw, we can measure the earliest one. There were no networks perturbed. In the second one, there was 1 network. In the third one, there were 2. And in the 5th one, there were 5 at least. And this is only a small subset of the networks that exist in that entity. And this means we have targets that we can go after again, either with immunotherapy or classical methods, or methods of a new approach that several of us are really hoping to pioneer here, of using peptides as drugs where you can make them large scale and very effective, but we haven't time to talk about that. And the detection of chronic disease through blood phenotypes and genetic risk, I think, is really an interesting idea. So what we did with the airfield population is we took them all and we looked at genetic risk, increasing genetic risk on the y-axis with regard to this paper published in Nature Genetics in 2018, where the y-axis was the phenotype of the disease. And what you saw with increasing genetic risk, a striking increase in disease features, okay? So what we decided to do was to replace the y-axis with blood analytes. What happened to each of 800 blood analytes as the genetic risk for 54 different diseases was interrogated? And in doing that, we found things that increased, things that decreased. We found in the 54 different possibilities of the order of 800 analytes that correlated up or down with these things. And for example, in the LDL cholesterol example, we saw a beautiful increase in LDL cholesterol with increasing genetic risk. And we demonstrated those at the highest genetic risk couldn't touch bringing the cholesterol down with exercise or diet. Those at the lowest risk with high cholesterol brought them right down. So you're going to be treating people with high risk and low risk in different ways, but it is not quite that simple. And I think this is really revealing. So this is a colleague of mine here at ISB who had 3 generations of males that died of heart attacks in their early 50s. He started taking his LDL cholesterol in his early 20s. And what he found, being young and fit, is in the mid-30s, all of a sudden, it shot up. And he was still young and fit. He still exercised. He still could. Something was programmed either by unknown personal behavior, the environment, or genetics to increase the cholesterol. Exercise and diet didn't touch it. Statins brought it right down. So again, we have to look at each individual and follow their analytes to look for these kind of transitions. And only then you have to worry about the high genetic risk. So if we do a lot of polygenic scores, the doctor should have them and the knowledge to know when they become a risk for the patient and what you can do for it. So the Phenome Initiative, there are really 3 things that we want to do: generate or aggregate large-scale phenome data, analyze it, and convert it into actionable possibilities and causal inferences. And the third thing I have down here is we want to identify partners that can help us translate this to patients, because we aren't going to become a hospital, anything like that. So we have data now, and it's probably 10 times as much data as we had for the Aerovale that we'll be collecting. And big differences are, one, we're going to do digital analyses of brain health. That's entirely missing in healthcare today. And we can talk about that with anyone that's interested. Number 2, we're going to take electronic health records and patient-reported data and environmental data and integrate those in as well. And point number 3, in selected cases, we want to do single-cell analysis of 5,000 white blood cells at each of the blood draws to be able to assess the relative balances of innate and adaptive immunity and the nature of the changes that actually occur in that regard. We've actually built a platform that is large scale, that can map into any of the clouds major clouds that exist here. It's built to receive knowledge graphs and a digital twin data in interesting ways. Modular and extensible, and it's to be used in conjunction. Well, we'll make it available to, frankly, anybody who wants to use it. We do have a spinoff now called Phenome AI that is going to use phenome data in the context of knowledge graphs, of digital twins, and ultimately of large language models. And without, I mean, there are 100 things you can do to this kind of approach. But what I'd like to see is that we create a system that can take the genome and phenome data from each individual and generate a prioritized list of actionable possibilities that will go to the physician with, 1, a description of it, and 2, its clinical evidence. And then 3, have something that come back to the patient in an understandable way with the appropriate treatment. And what this has the ability to do is to make, say, family practitioners domain experts in all areas of medicine and give them the ability to treat their patients as they've never, ever been treated before. One of the enormous challenges is how you persuade physicians to partner. And I think education is critical, and we can talk about that if you wish. So here are a list of short-term and long-term aims, and I'm just going to give you these very quickly so you understand. Initially, we're going to do as we did with Airveil. We're going to bring genome-phenome analyses to individuals. So we're going to collaborate with large healthcare systems and their family practitioners. And we have one candidate very interested and another just beginning to think about it. We have a commercial company that's very large that has enormous consumer connections that we're debating with now about whether we can apply this genome-phenome analysis. And of course, there are other possibilities too. We're going to take data-rich observational and interventional clinical trials for certain aspects of wellness and the 4 major chronic diseases. And of the chronic diseases we're starting with initially, we have the support to begin a 1,000-person study of diabetes, where we've selected the patients by electronic health records to span the entire life history of diabetes. So over a 4-year period, we can stitch that together very nicely. And we're setting up to do Alzheimer's as our next project. And in the wellness project we're really focusing on now is pregnancy. And we've already, together with Joel Zadowski at University of Pittsburgh, selected 460 women to follow through pregnancy at 5 different times with deep phenomics to analyze in enormous detail the placenta, and it has absolutely fascinating features. And then finally, we're going to try and raise the money to be able to follow mothers 2 years post-delivery and the children for the first 10 years of early childhood development. So we have complete records and so forth. Okay, we're going to look at interesting longitudinal banks, and we have a cohort in diabetes, a large government grant that allows us to do all the basic framework things, well, many of them, that we need for genome and phenome for analysis, and the spinoff company I talked about, and the long-term initiative, 10-year, million-person things. And I think it's going to do 3 things: prove improved health, identify and reverse. We'll begin to be able to reverse the transitions and catalyze the technologies that will allow us to create an impedance match between wearable entities and the omics, so we can take then and gather data in the home and thus democratize what we have here. So just, can we turn off the front lights? Or I guess probably that isn't possible. 2 days ago, I saw this spectacular accomplishment of the entire fly brain with connections and the neurons and so forth. And I would just make the point, deciphering the complexity of human health with the million-person project is going to be an equally large transition. And what it's going to enable us to do is deal with the issues of quality and aging and chronic diseases, equity, and exponentially increasing costs, as I've indicated throughout. And finally, we'll replace wellness and prevention for our current disease healthcare system. In democratizing healthcare, in time we can bring it to the entire globe, including underdeveloped countries. And finally, I think there's going to be a real revolution in AI. Is going to be one of the biggest transformations of medicine in the future. And the Phenome Health people have really done a magnificent job in putting this in place. And you can say, well, this sounds great. How are you going to do it as a small entity? And with funding, obviously we can grow, but the really key point is to identify superb partners who can provide resources, patients, and things like that that you need, and partner with them. So, thank you.